Question?
The attention-based morphogenesis that you're talking about there, the suggestion that you make that abnormal connectivity might lead to abnormal folding, do you have a sense of how that will then play into a disorder like schizophrenia, where we know there's a lot of differential sort of loss of grey matter? Is this sort of secondary to abnormal connectivity? We also know that grey matter loss has different trajectories for different sort of developmental courses of schizophrenia. Right?
So we are at the final stage of writing up a paper with John Cernansky where we've collaborated on the structural aspects of schizophrenia. We did find one folding abnormality in the parietal operculum region that was unexpected based on previous studies. So in that sense, it's a new discovery. I'll be quite honest, we were a little disappointed that we only found one and not 33. But you take what nature gives you, and it could be because of the diversity of schizophrenia. It's not a single disorder. Or it could be that it's just morphologically, whatever wiring is scrambled is not profoundly scrambled in the way that, say, appears to be done in Williams.
Or it could be that different developmental disorders have different sort of pathologies. One is caused by abnormal connectivity, the other one is about abnormal pruning. Or
it could be, yes. What I would say is I think, fMRI, task-based fMRI, and we're collaborating with Deanna Barch in the same schizophrenic population to look for functional abnormalities. And then this resting state functional connectivity, I'm an optimist. I think it's going to reveal some just surprising patterns of abnormal network, but it's not. There's evidence for it now. For example, Jessica Church in C. Peterson Brad Schlager's lab has shown resting state-based functional abnormalities in Tourette's syndrome that are different from what was expected but look pretty pronounced. So there's going to be a strong toolkit emerging here for these questions. Yes?
I'm curious how much, if any, of the cortical folding is influenced by some external factors. So a young child in the right kind of conditioning and the right kind of situations, will that have any kind of influence on folding, or is it mostly going to be just sort of standard?
You mean during postnatal development? Probably some. So I'm relatively neutral on this. I don't expect profound changes. There is some evidence that there are structural differences, say, in piano players compared to others in somatosensory cortex, and that could be an experience effect, or it could be they just have a folding pattern and a connectivity pattern that makes them good piano players. So the jury's out on that. What I can say is, from this collaboration with Jeff Neal and Terry Inder, we're going to have a very strong baseline for what cortex looks like in normal term infants as well as these preterm infants that are a combination of normal and abnormal. And we'll have some longitudinal studies to look at not just at the statistical average, but be able to follow cortical structure in individual children and to relate it to their developmental profile. So I think answers will emerge. So that pretty much the jury's out at this stage for me.
How applicable at the current time are the segmentation algorithms for postmortem brains or brains where there's been brain damage to a stroke or hemorrhage is present on the images, where you might then have a pathological study of some sort of cellular characteristics in an area where you had the brain at brain cutting over fixed or something.
So postmortem brains have been tough sledding until quite recently. Partly, a big factor is the contrast that you get looks just very different from a standard T1 weighted. Bruce Fischel has successfully segmented, using the FreeSurfer software, has successfully segmented a group of postmortem brains that came from Carl Zillis's lab, and so it can be done. And you also have the advantage in a postmortem brain, you can scan overnight if you want. They're not clamoring to get out of the scanner. And so if you're willing to pay the scan time, you can get higher resolution. The evolution of software algorithms, such as the one I showed, the preterm infants have a contrast closer to the postmortem pattern just because of coincidentally, I think. But those tools are probably going to help. So what has been rather difficult is going to become tractable and eventually, hopefully, easy. But
About clinical cases that may have
and
hardware in their head or blood or something.
yeah, and so Damien Fair in Steve Peterson's and Grad Schlager's group has done some segmentation of patients with large lesions, and one class of situations where you can reconstruct the hemisphere opposite to the lesion, but it may be deformed, and surface-based registration may help in getting alignment to an atlas better than volume-based. If you're dealing with a large lesion or even a modest-sized lesion, the cortex on that side has typically been topologically messed up. So we've been thinking about it and basically have been on the lookout for a test case that would have a clean enough lesion where we think we know what the answer should be, and we can actually explore ways to get a decent segmentation up to the edges of cortex and then try to figure out what to do. But because the topology is going to be messed up in a quasi-arbitrary way from case to case, it's kind of hard to figure out exactly how we will make progress on that. But it's a fascinating question. Yes?
Let's say if somebody has, let's say, a disease and, let's say, area 27, I don't know, 21, for example, and the physician wants to do some surgery or some effect on this, so does he or she needs to do an fMRI, or without fMRI, how they can find area 27, 21, for example?
Well, so this is neurosurgeons are getting very interested in using fMRI in the subjects, and there are a number of centers where fMRI is being used in those subjects. It's challenging because functional localization, even in normal subjects, remains tough. You know, most of the places, except for the early sensory topographically organized areas, getting clean maps of functional localization in a single subject is hard, even when they're healthy. But there will continue to be progress in cases where that is paying off. Our perspective on it is that we would like to bring the wealth of atlas-related data from many different subjects into the context of a clinical environment where it could guide a neurosurgeon to say this region is more likely to be language-related or tied to a particular function based on more effective use of the wealth of experimental data available. But if you're putting someone's life on the line, there are a lot of challenges that go beyond just making a best guess. And even though we think our tools have the potential to be more powerful, it's really you look at it differently if you're saying, is this going to guide a surgical decision? So we can maybe talk about that more later. Terry.
So in the X-ray crystallographic communities, for quite some time now, if you wanted to publish a crystal structure, you had to deposit your coordinates in GenBank, or not GenBank, but in I forgot what it's called. But in any case, a depository, so that anybody now could use those coordinates. Is there something similar to that happening for your database? In other words, obviously you're going to put your own papers in, but what's the incentive for other people to put their papers in your database?
So it's an excellent question. I would say the incentive becomes meaningful when the individual investigators say this is win-win. If people start discovering my paper because they found it in the database and then went to the paper, they'll be wanting to gain enhance their visibility through that mechanism. Unless and until that happens, the voluntary willingness to do the hard work and put the data in a usable format into a database is lower. The idea of requiring that TAILORAC coordinates be included, I don't know of a journal, maybe Human Brain Mapping requires it, but in general it's not a requirement. It's a common occurrence because people realize that's useful, but putting it into the database has been a bottleneck. And we have spent a large effort this past year particularly essentially trying to make the case this can be win-win, but it doesn't occur until you really make it get the infrastructure and the way of handling the metadata so it's a high percentage of signal, good metadata, and not a bunch of poorly characterized and annotated data. And that's where we're in the latter stages of trying to make that happen. But yes?
There's much more than me. I just got email sent to everyone at UCLA yesterday that NIH is requiring that all NIH-sponsored research put its data someplace.
Well, they are requiring that all publications be put on PubMed Central. There is a requirement related to data sharing that applies only to large grants. I've actually campaigned that I think it would be a good idea if every grant were required to address the issue of data sharing so that they do due diligence and say, is there a database where everyone, myself included, would benefit by putting my data there? But there's a reluctance to take that step yet because there's not enough successful proof that it really pays off. But I think that will change.